Maria calls the roll,
started.
right? Okay.
Yes. And we've got YouTube going, so I will call roll. Lauren Alving.
Here.
Mark Fisher Colbury.
Here.
Elena Flowers.
Present.
Judy Gasson.
Here.
Larry Goldstein.
Here.
David Higgins.
Here.
Shlomo Melmed.
Here.
Christine Miaskowski.
Good morning.
Jonathan Thomas.
Here.
Art Torres.
Present.
Carol Watson. Keith Yamamoto.
Here.
And then I would note that Deborah Deese and Pat Levitt are not present. So we can start.
Okay, great. First order of business is changes to the clinical plan advisory comments. I believe Alba Creasy is doing that one. Is that correct?
Abla Creasy is doing that. Yes. Thank you.
Oh,
Yes, good morning. Let's see if I can show my slides. Okay. Good morning, everyone, dear members of the Science Subcommittee. So I am the head of therapeutics development at CIRM, and I'm being today Gil, since he is on vacation this week and he usually gives these presentations. So I am presenting the proposed revisions to the CLIN2 concept. The proposed revision to CLIN2 concept is as follows. Therapeutic candidates currently allow clinical trial studies with a candidate that's either stem cell therapy or genetic therapy, phase 1, 2, or 3 clinical trials. While small molecule or biologics involve stem cells, phase 1 clinical trials only. We propose to unify eligibility to allow all 3 categories to qualify for phase 1, 2, or 3 trial. This action allows for consistent eligibility requirement across all clinical applications and provides the possibility of ongoing CIRM support for small molecules and biologics that are ready to advance to late-stage clinical development. The existing eligibility language would then be extended to phase 2 and phase 3 clinical trials. This is what you see on the slide. A small molecule or a biologic that acts on or is dependent on endogenous human stem cells for its therapeutic effect, that is dependent on targeting human cancer stem cells for its therapeutic effect, that modifies a stem cell therapy, or where a human stem cell is necessary to manufacture the therapy, such as extracellular vesicles. There are additional minor revisions that are being proposed mainly for clarification of the text, such as gene therapy to genetic therapy. There are also a few instances where the update was missed in the last round of changes to align with adopted definition of genetic therapy. Clarification that feasibility trials for medical devices are included within the requirements for award amount limits and co-funding amounts. So the action requested is to please approve of the proposed amendments to the CLIN2 concept plan. With that, I'll stop.
okay. Questions for Abla or other CIRM staff regarding this proposal?
Larry, why don't we get a motion on the table? I'll move.
Okay, thank you, JT. Second?
Second, David San Diego.
Okay, now then, discussion or questions, please. Let's see, Judy, did you just wave your hand?
Dr. Melmed has his hand raised.
Okay. Almost.
This sounds obviously very, very important and helpful to proceed with this. I'm just curious, being an old veteran on this committee, what was our original rationale for splitting it up? JD, do you remember, or was it even before your time? Why was this distinction made? Was there a reason for it which we're currently unaware of?
Can I answer that question? Well,
Sure.
Jacob, thank you.
Of course, Alba, please.
Okay, I think, again, as a newcomer to CIRM and having worked for pharma, I think the idea was beyond phase 1, it's likely that the pharma and biotech companies will invest in such a program. And it probably was one of the reasons, but maybe JT has others.
Yeah, the one of the big drivers, Shlomo, was when we were in running out of funds the couple of years prior to the passage of Prop 14, there was discussion about cutting back on certain things that were eligible for funding, and this was one of those. And so the idea here, I think, is to reinstate it because all of the phase 2 and phase 3 work, it all still must have to do in some capacity with stem cells one way or another, whether it's small molecules, biologics, or anything else.
Right.
So keeping that requirement in place, and now having the luxury, courtesy of the voters, of having the funding available, I think it was the team's view that it's time to expand the definition to include everything in phase 2 and phase 3 as well.
And towards the end of Proposition 71, we had limited funds, so we had to prioritize. And now that we have the Proposition 14, the funds could be made available if you decide to do that.
Let's see.
Larry, Maria
Yeah.
has her hand raised.
Got it. Maria Milan, please.
Absolutely. I mean, I just wanted to maybe give some context as to why we're bringing it to your attention now. It really came to our attention that, especially with the market as it is in terms of investment into early stage programs, and some of the programs we had funded through the early stages into phase 1 that showed some promise, including combination therapies where small molecules or antibody or biologics are needed for the success of cell transplant, like non-toxic conditioning regimen, for instance, or some of the novel small molecules and biologics that targeted a stem cell regenerative medicine mechanism of action that is not necessarily out in the standard development path, or the types of approaches that address rare disease, ultra rare disease, which currently right now are being taken care of by academia. These are the types of programs that are uniquely the types of programs that wouldn't have even gotten this far were it not for CIRM. And they also would have had a hard time getting industry support, even under the best circumstances, but especially with a market where it is and investments are access to capital is very challenging, very competitive right now. We wanted to make sure that programs that we had invested in, and especially those that could be very impactful to the mission and to the patients and to the types of technologies we're bringing forward, that we have a way to take care of those programs. So that's the context of why today, in addition to the fact that we also have as JT and Abla had pointed out, now we have the ability to support it through the renewed funding.
Good. Now, so I was just concerned there was a policy issue, but based on what you said now, we have to, and I applaud this approach, and it's great that we're doing this, but we have to realize we're in for hundreds of millions of dollars. I mean, a successful phase 3 trial, if it's well designed, is extremely expensive.
But our funding will continue to be the same, meaning we will, for example, a phase 3 trial will get $15 million from us. And the rest will be through co-funding and other resources that the party would have to identify. The key here also is please remember that therapeutics, whether small molecules and biologics, will have to have some relationship involvement with stem cells. So those are the ones we're advocating for.
Thank you.
Other questions from members of the subcommittee? Okay, hearing none. Let's see, Maria, is this time for public comment?
It is, and I do not see any hands raised for public comment.
Okay, so
Nope.
then I suggest, let's see, do we have to have another motion to vote or can we go straight to
No,
vote? Maria?
We can go straight to the vote.
Okay. Call the question and vote, please.
Lauren Alving,
Yes.
Mark Fisher Colbury,
Yes.
Elena Flowers,
Yes.
Judy Gasson,
Yes.
Larry Goldstein,
Yes.
David Higgins,
An enthusiastic yes.
Shlomo Melmed,
Yes.
Christine Miskowski,
Yes.
Jonathan Thomas,
Yes.
Art Torres,
Aye.
Carol Watson,
Yes.
Keith Yamamoto.
Yes.
The motion carries.
Okay, thank you, everybody.
Good.
Next up is Sham telling us about a proposed concept for a network of cell and gene manufacturing facilities. Sham, take it away.
Thank you, Dr. Goldstein. So I'm going to put up my presentation. 1 second, please. Sorry, 1 second. It's just transferred to a new laptop and having some permission issues. So trying to work it out.
Tom, do you want us to share the screen with what's posted online? You just let us know.
I guess we could go with that because this is not working at the moment.
Marianne, do you want to grab, do you want to share the document that Sham sent you that we posted?
One second. Got it.
Thank you, Marianne, and I'll just let you know when to progress slides. So I apologize for that. Today I'm going to be presenting a concept plan for a manufacturing network in the state of California for cell and gene therapy manufacturing. I'm going to say in advance that there were supposed to be quite a few animations in the slides, so I'll do my best here with respect to walking you through a lot of text. So just bear with me, please. So as we always start off with the mission statement, accelerating world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. So next slide, please. Late last year, the board had approved our 5-year strategic plan. And within that plan, there were 3 major themes. And this particular concept plan addresses one of the objectives in the Deliver Real World Solutions theme, which was creating a manufacturing partnership network. But it also touches on the competency hubs and the workforce development objectives of the other 2 themes. And I'll be walking through the presentation, the concept plan over the next few slides. Before I do that, I want to set the background in terms of the current bottlenecks in manufacturing. So next slide, please, Marianne. So we're all aware that there's been a rapid growth in regenerative medicine, particularly in cell and gene therapies, and now they are rapidly progressing through clinical trials in many instances. And that rapid progression of these therapeutic candidates often creates a burden on manufacturing, where some of the bottlenecks in manufacturing development can actually slow down the overall development of the therapies themselves. And so there are a few infrastructure bottlenecks as well as some technical bottlenecks and resource bottlenecks that are addressed here. So first and foremost, the academic institutions are the center of technology innovation, initial process development, and GMP manufacturing, but they don't have sufficient capacity, resources, or processes for late-stage manufacturing, and often this is by design. On the industry side, there are industry contract development and manufacturing organizations that specialize in manufacturing for sponsors, or in-house manufacturing operations of companies of all sizes. And these are best positioned to industrialize manufacturing processes for late-stage clinical trials and commercialization, but they don't always have the expertise in emerging technology platforms that arise from academia, and which is often the case for cell and gene therapies in particular. So on top of that, there's the technical bottlenecks that arise from the complexities of their products and processes themselves. These are living products in many cases, and they have complicated multi-step processes. And then lastly, because of the rapid growth in the industry, there's been an ever-growing demand for trained manufacturing and quality workforce. And so some of these areas are being addressed by the concept plan being presented to you today. So first and foremost, I want to lay the landscape. So next slide, please, Marianne. So in California, we benefit from having a very rich network of academic GMP manufacturing facilities. In fact, a majority of CIRM-funded projects utilize these GMP manufacturing facilities for the cell and gene therapy product development. These include facilities that have been around for a long time, like the UC Davis, UC San Diego, and City of Hope, as well as recent ones that have just opened, for example, Cedars-Sinai, UC Irvine, and USC. On the industry side, there has been a growing presence of contract development and manufacturing organizations. These either provide fee-for-service or have partnership models. There's been a rapid growth of these, especially in the last couple of years. And prior to that, there really weren't that many industry CDMOs in the state of California. In addition to that, several biopharma companies are also manufacturing in California for their partner projects. And two of our industry alliance program partners, Bayer and Novo Nordisk, both have this capacity at their Bay Area facilities at the moment for partner projects. Next slide, please, Marianne. So given that landscape, in the strategic plan, we had proposed to create a manufacturing network that links the advantages of the academic GMP facilities with the advantages of the industry partners, all in service of three core goals. The first would be to accelerate and de-risk the pathway to commercialization for CIRM-funded cell and gene therapy projects. Second, to advance standards and quality by design. So these are industry standards that would help industrialize the process and create more consistency and higher quality products. And lastly, to build the manufacturing leadership and workforce in the state of California. So next slide, please, Marianne. What could be some of the potential functions of the manufacturing network to address those three goals? And those are described in this slide here. So first and foremost, world-class expertise across the full range of manufacturing and analytical technology platforms, from pluripotent stem cells to gene editing, for example, to support the manufacturing of therapies for rare, ultra-rare diseases. These are platform-based approaches that could potentially develop and manufacture variants of cell and gene therapy products for a large number of ultra-rare diseases. To accelerate and de-risk late-stage and commercial manufacturing of therapies. This is particularly important as the field matures and as does CIRM's portfolio, and the needs for late-stage manufacturing for larger clinical trials, as well as commercial manufacturing for approved therapies, increases in the state of California. To establish standards for quality or accreditation of manufacturing facilities, this is equally important as the field matures. And lastly, to build inclusive workforce entry and advancement opportunities in technical and leadership career pathways that partner with our EDUC programs, as well as industry stakeholders ranging from community colleges to biotech companies to the contract manufacturers in the state, as I listed previously. So the next few slides are going to walk through the concept plan. This is a broad overview first, and then potential activities. So this is a biphasic funding opportunity that we're presenting in this concept plan, composed of two distinct but interrelated RFAs. So the first phase of the RFA would be have a program budget of $20 million. These would be max 2-year, max $2 million awards. And the applicant and awardee would be academic GMP manufacturing facilities, with the intent that these phase 1 awards are focused on individual facility enhancements at those facilities themselves. The phase 2 RFA would have a program budget of $60 million with a max award duration of 5 years and a max award amount of $5 million. Here, the applicant GMP facility would be applying to propose collaborations, which in turn would allow for scaling of the enhancements and specializations and training programs that they had made progress toward in the phase 1 application. So basically the phase 2 is scaling up in collaboration with others of the activities that they've done in phase 1. So in addition to the awards themselves, CIRM would coordinate a steering committee of awardees and external participants. This steering committee would play an important role, which would act as the glue between the awardees themselves, as well as to facilitate the transition from phase 1 to phase 2. So in the next few slides, I'm going to walk through potential activities for phase 1 and phase 2, how phase 1 and phase 2 interplay with each other, as well as what the steering committee itself could be doing. But I do want to note that both in the phase 1 and phase 2 RFAs, the encouragement and expectation is that the awardees are working in collaboration with other academic facilities, as well as industry stakeholders in the state of California. So this is a very busy slide because it was meant to progress through some of these things. So just bear with me as I walk through this. So the potential award activities that we envision for both phase 1 and phase 2. Again, these are meant to be examples as informative information only and not meant to be limiting in any way for the eventual RFA and awards, are bucketed into 3 major categories. So first and foremost is to de-risk and accelerate manufacturing, a theme that we've been constantly talking about throughout this presentation. And so here, potential activities could involve quality-driven operational enhancements that de-risk process development, GMP manufacturing, and technology transfer from pre-IND through to commercialization. It could involve actively mitigating capacity and expertise gaps by coordinating project execution across the network. Oftentimes, some projects may have lead times of a couple of years, or some facilities may not have the expertise in a particular area, but they could coordinate with others to support those projects. We would encourage applicants to propose specialization in particular areas. So these could be building network-wide specialization in areas such as technology platforms, CRISPR, for example, analytical methods, pioneering quality by design implementation, automation of manufacturing, or having rare disease manufacturing platforms. And lastly, on the workforce development side, they would be proposing development and implementation of training programs for technical positions that could involve internships and certification programs, as well as mentoring programs for leadership positions. And these would be in partnership with our EDUC programs, which could be the feeder for a lot of these positions, as well as with industry stakeholders, which could be both the feeder and destination for some of the trainees. So as I mentioned previously, the phase 1 awards would focus on initial progress in these areas at the academic facilities, while phase 2 would be how to scale these across the network. And so what I'm going to do is to illustrate that interplay between phase 1 and phase 2 is to walk you through potential activities that could be done in phase 1 and phase 2 for that first example, which is outlined in that box around de-risking manufacturing. Next slide, please, Marianne. So here on the phase 1 side, to de-risk manufacturing, those individual awardees could be making quality system improvements at their facilities. They could be implementing quality by design principles, and they could be hiring and training new staff around that to basically bolster their operations, all with the intent of being able to better support process development, to better tech transfer projects into their facilities for early stage manufacturing, and then to better transition those projects out of their facilities for late stage manufacturing when the need arises for those projects for later stage clinical trials, as well as commercial manufacturing. So a potential outcome metric for a phase 1 award for this particular set of activities could be how well did those quality system improvements impact project execution compared to historical performance at those facilities or globally. And then to tie that into the phase 2, Now, based on knowing which quality-driven improvements were successful, those could be scaled across the network and across the participant or organizations. And then on and they could also operationalize partnerships to effectively transition projects for late-stage commercial manufacturing. So based on what are the best practices in terms of coordination between academic facilities and industry partners to facilitate progression of projects from initial manufacturing to late-stage manufacturing, those could be operationalized more fully in the phase 2 awards. So potential outcome metrics for phase 2 awards could be what is the success rate of transitioning projects to late-stage manufacturing, as well as how well were the quality standards, protocols, and best practices applied across the network. So to go back to the other 2 categories and propose some outcome metrics for those. So with respect to specialization areas in phase 1, the awardees could demonstrate competency in specialization areas of executing pilot projects. And then in phase 2, they could demonstrate how effectively were those specializations utilized by the other facilities in the network. And with respect to the workforce development, in phase 1, the awardees may demonstrate the enrollment of the first trainee cohorts for both the technical and leadership training programs. And then in phase 2, they could demonstrate successful enrollment in training programs, as well as the success rate of trainee job placement. And one thing I should note, as I've been talking about these activities and potential outcome metrics, is that the funding in these awards will be to the facilities around operational enhancements, but the actual projects that they're supporting for cell and gene therapy manufacturing, the funding from those would still flow from the Tran and Clin awards and will be controlled by the sponsors themselves. So that the actual project support is coming from our pipeline programs, but this funding mechanism and the funding here is focused with the operational enhancements being made at the facilities. In that sense, it's pretty analogous to the Alpha Stem Cell Clinic's funding opportunities. So next slide, please. All of our current funding opportunities address DEI as well as knowledge sharing. So I'm just going to briefly walk through how both of those components are addressed in this particular concept plan. So with respect to diversity, equity, and inclusion, the applicants must propose a plan to demonstrate how their training programs in particular will ensure participation by underserved populations, as well as how their project teams represent diverse and inclusive perspectives and experiences. Next slide, please. On the knowledge sharing side, the applications will include knowledge sharing plans that describe the plan to capture and disseminate relevant know-how, operational data, processes, expertise, and guidance within the network. So this is all the knowledge sharing component within the network across the different participants that will really be the collaborative glue of the concept plan. Second is to describe any knowledge sharing plans that are critical to achieving the award objectives. And lastly, is to describe how their facilities data management processes will support CIRM, Tran, and Clin awardees to execute on their respective data management and sharing plans, which are now required for all Tran and Clin awards going forward. And so I mentioned the CIRM Coordinated Steering Committee. So I'm going to describe in this slide some of the functions that the steering committee could potentially do, and the next slide, how they tie into the activities themselves. So CIRM will coordinate a steering committee of awardees, California industry partners, as well as a rotating group of national stakeholders to facilitate identification and adoption of standards, protocols, and best practices across the network, and to create potential criteria for facility accreditation, to mitigate capacity and expertise gaps across participating sites, to facilitate collaborative planning for phase 2 proposals between the academic and industry participants, to develop systems and processes for sharing information and resources between network participants, and lastly, to collaborate, to promote collaborative development and implementation of workforce training programs. On the next slide, I'll describe how the steering committee might play a role on the activities I previously described a few slides ago. So with respect to de-risking manufacturing, what could be the role of the steering committee? With respect to the phase 1 awards, the steering committee could identify quality standards for academic GMP facilities to adopt. They could also define the knowledge sharing processes that may be required between the facilities to help facilitate best practices sharing. On the phase 2 side, the steering committee could apply the quality standards consistency across the network. They could facilitate knowledge sharing within the network through systems and processes that allow for that. And lastly, they could actively triage projects by expertise and capacity across the network. And then on the last slide, I'm going to describe the award information. Some of this is information I presented previously. So the overall program budget is $80 million. Phase 1 is $20 million, which would go live this year if it's approved by the board. And then phase 2 will be down the road and would be $60 million. The awards themselves would have caps of $2 million for phase 1 and $5 million for phase 2. The allowable costs for these awards include direct project costs and direct facilities costs. They don't include indirect costs, which is consistent with our other infrastructure funding opportunities. And they will also include a co-funding component, which will be required for both phases of the awards at 20%. In terms of the applicants, just reiterating that the California nonprofit GMP manufacturing facilities can apply, and they must have a track record of cell and gene therapy project support. And that's the end of my presentation. So CIRM asked the board to approve this concept plan, and I'm happy to take any questions that you may have.
Okay, let's see if I can get it right this time. Is there a motion to approve?
So moved.
That was David Hagan.
Second, Chris.
Where
Thank
did
you.
the second- You got that,
Christine
Amanda?
Miscast.
Ah,
Yes.
good. Okay. Great. Art, you have a question?
You're on mute, Art.
Thank you, Shan, for that excellent presentation. I just had a question. What is considered a nonprofit GMG?
That's a good question. So it would mostly be the academic facilities, academic institutions that have GMP facilities, the ones I listed on the slide. So those would be the UC, City of Hope, Stanford, Cedars-Sinai, and so on.
So why did you exclude industry?
So the reason for that is that a majority of our projects go through the academic GMP facilities at the early stages. And so here the intent is that if we start them off on the right track in the first place, that the late stage transition to industry partners could be smoothened. In addition to that, we thought that where the funding is most critical is for the academic partners, and the industry partners could be the ones who are collaborating and providing their resources here and being the eventual house for those later stage projects.
Thank you very much. Thank you, Mr. Chairman.
Other questions or comments? You know, I actually have JT, go ahead. You have your hand up.
So Sean, when was the manufacturing workshop?
So the manufacturing workshop, and I apologize, all the timing is blended in my head, but I believe it was last year. Or it could have been the year before.
Yes. No, it was last year. It was last year, Sean.
It was earlier last year. So I just raised this just to make the point that the thinking on the manufacturing process and what we can do to be a player in that is involved a tremendous amount of work over many, many months. Just the planning for the manufacturing workshop and getting the attendees and everything that went into that was a huge amount of work. And all of it that's come subsequently was a function of the lessons that were derived from that workshop, and it ultimately culminates in this concept plan, which, as you can see from its complexity and thoroughness, is involved a huge amount of work. So I say all of this just to congratulate Sham and all members of the team for putting together a first-rate concept plan that will dramatically improve the manufacturing environment in California, and as a result, for patients everywhere. So just a comment to that effect. Very well done.
Other questions or comments? Sham, I have a minor suggestion, which is you might want to make it clear that this is not including chemical synthesis or antibody production, that it's limited to production of cells and probably viral or other types of vectors.
Thank you, Larry. As always, your recommendations are spot on.
Thank you. Other comments or questions? Public comment. Anybody on the line, Maria?
If there's any public comment, if you could please raise your hand, and then you would have 3 minutes to speak. I do not see any public comment.
Okay, hearing none. Oh,
Oh, I'm sorry. Zach, you have 3 minutes.
Hi, yeah, I'm not so familiar with these procedures. I'm just wondering who is responsible for tracking the outcome metrics.
Thank you. That's a good question. So these will be built into the awards themselves. And so the metrics will be, so that whatever outcome criteria we establish as part of the milestoning of the awards would be tracked by the awardees and then evaluated by CIRM science officers.
Thank you.
Larry, we have one more person
yes.
for public comment.
Please.
Yes.
Yeah, hi, thank you guys. This is a great concept plan. I have a question regarding the phase 1 part of the proposal where there is some description regarding specialized offerings that, you know, are sort of supposed to fund possibly technology development platforms like CRISPR or iPS cells or automation, etc. Is it, so I assume then this is something that the academic GMPs will be developing independently of a client's project, or a particular for-profit client or not-for-profit client that's already ongoing in the GMP facility. Can they leverage that to be part of this proposal, or are they mutually exclusive? It'd be helpful to clarify that.
Thank you, Drew. Quick question. So with respect to that, yeah, so it could be that there could, you might have to do some development independent of what is being funded through a particular project. And then you could demonstrate that you've made that specialization with that project. So it doesn't specifically have to be completely independent. They could be related. And this funding mechanism could be used to create, to do some of the development that you might want to do independently of what might be contracted through that project.
Okay, thank you.
Thank you for that comment and question. Let's see, anybody else? Okay, if not, then we vote.
Lauren Alving,
Yes.
Mark Fisher Colbury,
Yes.
Elena Flowers,
Yes.
Judy Gaston,
Yes.
Larry Goldstein,
Yes.
David Higgins,
Yes.
Shlomo Melmed,
Yes.
Christine Miskowski,
Yes.
Jonathan Thomas,
Yes.
Art Torres,
Aye.
Carol Watson,
Yes.
Keith Yamamoto.
Yes.
The motion carries.
Okay, thank you, everybody. Any final comments or questions? We've pretty much reached the end of our agenda. Any other public comment not related to this topic? Nothing going.
Nope.
Okay, with that, I suggest that we adjourn. See you at the next meeting. Thank you all for your time.
Thanks, everyone.
Thank you.